Hello everyone. Today we will learn about neuroanatomy. Under neuroanatomy, the first topic is organization of nervous system, where we are going to learn basics of neuroanatomy. Nervous system is meant for the perception of internal and external environment, which respond with appropriate reactions produced by the muscles, organs and glands. The human nervous system is the most complex and versatile creation of evolution, with advanced functions like cognition, learning, memory, intellectuality, personality and many more. So here is the picture of a nervous system where we can broadly divide into central nervous system, which is formed by brain and spinal cord, and peripheral nervous system formed by the cranial nerves which are arising from the brain and spinal nerves which are arising from the spinal cord, and autonomic nervous system. Let's see little more in detail. Here is the picture of the central nervous system where we can see the brain here. This is the brain, and brain continues below as the spinal cord. So, the caudal extension of the brain is the spinal cord. And if we take a sagittal section from the midway of the brain, dividing into two halves, we can see little more in detail. So, here is the sagittal section of central nervous system showing the brain again, and spinal cord present within the vertebral column. Here is the sagittal section of the brain. Now we shall see the parts of brain, components of brain. So it is mainly formed by the cerebrum, which is the largest part of brain, midbrain, pons, this is cerebellum, which is also called as little brain. So these are the parts of brain. Here is the autonomic nervous system. Autonomic nervous system is further can be classified into two parts. One is the parasympathetic, the other is the sympathetic. So this is the sympathetic component of autonomic nervous system. The sympathetic component is also called as thoraco-lumbar outflow. Thoraco-lumbar outflow, because it is arising the nerves which relay in the sympathetic chain, which is present on each side of the vertebral column and arising from the spinal cord, preganglionic fibers and postganglionic fibers from the sympathetic chain. They go to various parts of the body. This is parasympathetic nervous system. Parasympathetic nervous system is also known as cranio-sacral outflow. So, the cranial nerves which are arising from the brain and the nerves which are arising from the sacral region, they form the parasympathetic nervous system. We saw the sympathetic ganglions near to the central nervous system which are present on each side of the vertebral column or spinal cord, whereas the parasympathetic ganglion are present near the target organs. So, here are the parasympathetic ganglions which are present near the target organs. So, these two are the components of autonomic nervous system. Peripheral nervous system also consists of cranial nerves and spinal nerves. There are 12 pairs of cranial nerves and 31 pairs of spinal nerves. Here we can see the picture showing the base of the brain, where the 12 pairs of cranial nerves which arise from the base of the brain, and they are numbered from anterior to posterior. So, from the first I am just writing numbers. The first cranial nerve is the olfactory nerve. This is olfactory. The second cranial nerve is the optic nerve. Third cranial nerve is oculomotor, which arises from the midbrain. Fourth cranial nerve is also present along with the oculomotor, arising from the midbrain. The fourth cranial nerve is trochlear. Fifth cranial nerve is the thickest among all, which arises from the pons, called as trigeminal nerve. Sixth cranial nerve, it is present between the pons and medulla here. This is the sixth cranial, which is called as abducens nerve. Seventh cranial nerve, which is little lateral to sixth, is the seventh facial nerve. Eighth cranial nerve is called as vestibulocochlear nerve. Ninth cranial nerve is glossopharyngeal nerve. Ninth cranial nerve arises from the medulla. Ninth is glossopharyngeal nerve. 10th cranial nerve lies just below the 9th, that is vagus. 10th cranial nerve. And 11th cranial nerve, I am writing on the other side, 11th cranial nerve, which is the spinal accessory nerve. And 12th cranial nerve also arises from the medulla, which is called as hypoglossal nerve. So, these are the cranial nerves which are numbered from anterior to posterior in the base of the brain. Let's see the spinal nerves. Spinal nerves arise from the spinal cord, and there are 31 pairs of spinal nerves. So, the first 8 pairs are called as cervical, which are getting origin from the cervical region, are called as cervical nerves. So, first 8 pairs are cervical. Then 12 pairs are thoracic, then 5 pairs are lumbar, five pairs are sacral, and the last one pair is the coccygeal, which is also present along with the sacral nerves, coccygeal. So, these are the spinal nerves which are arising from the spinal cord. There are 31 pairs of spinal nerves, cervical 8 pairs, 12 thoracic, 12 pairs are thoracic, lumbar 5, sacral 5 and coccygeal 1. And next about the cells of nervous system. Nervous system consists of two varieties of cells called as neurons and neuroglial cells. Neurons, the basic structural and functional unit of nervous system, is the nerve cell or neurons. And these cells conduct the impulses and have the unique properties of reception, integration, interpretation and transmission of impulses, means the information to the target organs for appropriate response. So, there are approximately around 10 to the power of 10 to 10 to the power of 12 neurons in our human brain. So many neurons are there, and these neurons do not undergo mitosis after birth. So, they won't undergo cell division. So, the number of neurons are fixed after birth till death. And these neurons, here is the structure of a typical neuron showing the cell body. Cell body is also called as perikaryon or soma, and it consists of several processes which are called as dendrites and axon. The neuron have a specific staining towards Nissl stain because it consists of some substances in the cytoplasm, and these substances are called as Nissl bodies. These substances are called as Nissl bodies, which have affinity towards the Nissl stain. What are these Nissl bodies? Nissl bodies are rough endoplasmic reticulum of neurons and consists of granular bodies in the nerve cell bodies. and dendrites. So these cell bodies are present in the cytoplasm of the cell body as well as in the processes called as dendrites. And any nerve injury, the nissel bodies will degrade. So degradation of nissel bodies occurs, which is called as chromatolysis during any nerve injury. A typical neuron have a variable number of short branching processes which are called as dendrites. So these are short branching processes. As they divide, the thickness of the dendrites reduced, and the dendrites carry the impulses towards the cell body. So they receive the impulses towards the cell body, so they are afferent, and they connect with the other neurons by synapse. And these dendrites are supported by the cytoskeleton called as microfilaments. So the cytoplasm present inside the dendrites consists of microfilaments. And one among the process we can say it is quite longer, which is called as axon. And the axon is a longer process which is a projection from the neuron cell body, and they carry the impulses away from the cell body. So they are efferent. Dendrites are afferent. And axon is connected to the cell body or the pericardion or soma by this widened structure called as axon hillock. Axon hillock is the trigger zone for the initiation of action potentials, means the stimuli gets initiated from the axon hillock, and axon hillock lacks of this rough endoplasmic reticulum, and it also devoid of nissel granules and nissel bodies. And axon is also devoid of nissel granules and nissel bodies. The cytoskeleton of the axon consists of microtubules and neurofilaments. Microtubules means tube-like structures, filaments means thread-like structures. Microtubules are associated with motor proteins, two varieties of motor proteins called as kinesin and dynein. So axons are associated with two types of motor proteins called as kinesin and dynein. Kinesin, the motor proteins, they move from negative to positive charge, means from minus to plus. Whereas dynein moves from positive to negative charge. So this movement from negative to positive is called as antegrade transport, and dynein, the movement from positive to negative, is called as retrograde transport. This is the structure of neuron, where we can see the retrograde and antegrade transport from the axon. Antegrade is away from the cell, retrograde is towards the cell body. So antegrade is from negative to positive, retrograde is from positive to negative. So that is about the kinesin and dynein. The brain and the spinal cord mainly consists of two types of matter called as grey matter and white matter. Grey matter consists of these nerve cell bodies. So nerve cell bodies are located in the grey matter, and the group of these nerve cell bodies within the CNS are called as nuclei, and the group of nerve cell bodies outside the CNS, that is outside the central nervous system, in the peripheral nervous system, are called as ganglia. Let's classify the neurons based upon their structure. Here is first classification based upon the polarity. Polarity is the orientation based upon the number of poles from where the dendrites or axon emerges. So multipolar neuron. Here is the multipolar neuron having dendrites all around, and we can see a single axon arising from one side. So the multipolar neuron, we can see the pericardion gives rise to multiple number of dendrites and a single axon, and which is like a typical neuron, and example motor neurons. Next is the bipolar neuron. Bipolar neuron have two processes. One is the dendrite, the other is the axon. So here is the dendrite, this is the dendrite, and the other side is the axon. This is axon. And bipolar neurons are present in olfactory epithelium, nasal cavity, retina, the eyeball, and ganglionic cells of the cochlea and vestibular nerves present in the internal ear. The third variety is the pseudo-unipolar neurons. Pseudo-unipolar means false, and they have a single process which emerges from the cell body. So this is a pseudo-unipolar neuron which is having a single process, and immediately this single process will split into ascending and descending fibers. One is called as the dendrite, the other is called as the axon. So it is called pseudo-unipolar because during development they are like bipolar, having two separate processes. Later, during differentiation, the two processes approximate with each other to form a single process for a short course. And this kind of pseudo-unipolar neurons are present in the dorsal root ganglion. The last variety is the unipolar neurons and has a single process which terminally divides into dendrites and axons. So here is the unipolar neuron which terminally divides into dendrite and axons. And these are unipolar from the beginning of the development. So that is the difference between unipolar and pseudo-unipolar. And example, mesencephalic nucleus of trigeminal nerve. So now let us classify the neurons based upon the relative length of axons and dendrites. So based on the length, there are two varieties, Golgi type 1 and Golgi type 2 neurons. Golgi type 1 nerve cells are having long axons which may extend up to 1 meter length. And here is a Golgi type 1 which may extend up to 1 meter length. Example, pyramidal cells or bed cells or anterior horn cells arising from the spinal cord. Pyramidal cells and bed cells are present in the cerebrum. Golgi type 2 neurons, axons of these neurons are short, similar to the length of dendrites. So they are very short, like similar to the length of dendrites. So you cannot differentiate axons as such. And these kind of cells are present in the cerebral cortex and cerebellar cortex. So this is an example of Golgi type 2 neuron. And physiological classification of neurons, we can classify them as sensory neurons and motor neurons. Sensory neurons are afferent, means they carry the impulses from the receptor organs to the central nervous system. So they carry the impulses from the receptors to the central nervous system are the sensory neurons. And motor neurons carry the impulses away from the central nervous system, so they are efferent. So these are motor neurons which carry the impulses away from the central nervous system. So that is all about the neurons, and next we shall start with the neuroglial cells, which are the other type of cells present in the central nervous system. And neuroglial cells are supporting cells or interstitial cells which are present in between the neurons and which can regenerate, means they have the capacity to divide and form the major part of nervous system. And these cells are non-excitable cells, so they do not contribute in the propagation of impulses, means they don't transform the information. So they are just present around the neurons as supporting cells. And neuroglial cells are present in 4 varieties in central nervous system. The 4 types of neuroglial cells are astrocytes, which are star-like, that is the reason they are called as astrocytes. These green colored ones are astrocytes. Oligodendrocytes, oligo means few, dendrocytes means processes. So these are the cells, neuroglial cells, which have few number of processes. They are called oligodendrocytes. The blue ones are oligodendrocytes. Ependymal cells, they are like columnar cells with the cilia. they are lining the ventricular system of the brain. So, these purple cells are ependymal cells. And microglia. Micro means small. They are small cells. So, they are called as microglial cells. So, these are microglia. So, these are the 4 varieties of glial cells or neuroglial cells. We can say glial cells also. Astrocytes are the largest and most numerous among the neuroglial cells. Astro means star-like. They fill up the extracellular spaces in between the neurons. Astrocytes provide physical support to the CNS. They are like scaffolding for the CNS, and they provide extracellular potassium buffer. And these cells remove the excess neurotransmitters which are produced by the neurons, and they function as glycogen reserve. They contain bundle of intermediate filaments, and these filaments are made up of glial fibrillary acidic protein GFAP. Glial fibrillary acidic protein, which is an astrocyte marker. And these astrocytes proliferate and become hypertrophic if there is any CNS injury. So, they are involved in the nervous tissue repair. Astrocytes form a scar in reactive gliosis, and cell bodies are star-like and they have like dendrite process. So, cell bodies are star-like, having a many dendritic process all around the cell body. So, the end of the astrocyte is like a small swelling which is like a foot. So, it is called as foot plate of one end and in contact with the capillary. The other end is also forms a foot plate where it is in contact with the cytoplasmic membrane of the neuron. Thus, the neuron is not in direct contact with the blood capillaries. So, the capillary wall, the endothelial lining, here we can see the endothelial lining which is enlarged and shown here, endothelial lining, and we can see beautifully the foot plates of astrocytes forming blood-brain barrier. Blood-brain barrier means they allow only particular substances into the neurons. So, they act as a blood-brain barrier because they allow only particular substances can pass through the blood to the neurons. One more thing about these foot plates of astrocytes, these foot plates are quite dense and they make a resilient membrane forming a true capsule of the brain and spinal cord. And they are composed of processes of astrocytes, means microglial cells, and covered throughout by the matter. So, they firmly adhere to it, and the two membranes are collectively called as glial membrane. Astrocytes can be classified into again 2 varieties, protoplasmic astrocytes and fibrous astrocytes. Protoplasmic astrocytes are mainly present in the grey matter and their processes are thick and branched and tubular. So, they are branched, thick and tubular like. So, they are called as protoplasmic. In contrast, the fibrous astrocytes are found in the white matter and their processes are thin fiber like and less branched. So, I am drawing for you all. So, they are like thin fiber like and less branched, having the nucleus. So, these are fibrous astrocytes. Protoplasmic astrocytes will have thick tubular processes which further branch. These are protoplasmic astrocytes. Protoplasmic astrocytes are present in the grey matter and fibrous astrocytes are present in the white matter. Next, about the oligodendrocytes. Oligo means fewer, dendrocytes means processes. These are the neuroglial cells which have fewer number compared to astrocytes. They have fewer number of processes and they form the myelin sheath of axons in central nervous system. We can see it is forming the myelin sheath of axons of central nervous system, and the myelin sheath not only in the central nervous system, it also forms the myelin sheath of cranial nerve second. The second one is the optic nerve. So, it forms the myelin sheath of optic nerve too, and these cells develop from the neuroepithelium, that is from the neuroectoderm. Each projection can myelinate. We can see here in this picture, it is a beautiful picture showing that it can myelinate more than one here, but actually it can myelinate more than 30 to up to 60 axons. And it appears to be like a fried egg under histological view, and which is a prominent nucleus with a clear and pale cytoplasm. The third variety of neuroglial cells are ependymal cells, which are also called ependymocytes, and these are the choroid epithelial cells which line the cavity of brain and spinal cord, and they are also present in the choroid plexus, and they are simple columnar cells. They are simple columnar cells. Here we can see in purple color, and apical part of the cells shows cilia facing the cavity, lining the cavity, and this cilia helps in the circulation of CSF, cerebrospinal fluid, and the microvilli increase the capacity of CSF absorption. And these modified ependymal cells known as choroid plexus, which produce the cerebrospinal fluid. Tanycytes. Tanycytes are the type of ependymal cells which are in contact with the blood vessels, and their main function is to transport the substances between the blood and the ventricle. So, they are also modified ependymal cells. Now, the last variety of the glial cells are microglial cells. Micro means small. So, they are the smallest among all the glial cells, and these cells in the CNS, they are derived from the mesoderm. Means the whole nervous system is derived from the neuroectoderm except these cells which are derived from the mesoderm, and they are smallest among all, and they have the capacity to migrate into the surrounding neuronal tissue and become phagocytic when needed. So, here are the microglial cells which are the smallest among all, and these phagocytic cells, they wherever there is an inflammation, they engulf the pathogens, waste products and cell debris, and capable of releasing an inflammatory mediators and signaling molecules like nitric oxide, glutamate respectively. And these are the target cells of HIV virus. Means the infected cells fuse to form the multinucleated giant cells, which are more specific in histological marker of HIV encephalitis and HIV associated dementia. Microglial cells are poorly identified with Nissl stain. So, Nissl stain, already we heard about it in the neurons, where it is a chrysal violet stain, and it is an aniline dye which stains the RNA blue and has the affinity for polyribosomes, rough endoplasmic reticulum present in the cytoplasm, called as Nissl substance and Nissl bodies. Let's talk about the neuroglial cells of peripheral nervous system, and there are two types of neuroglial cells in PNS, that is satellite cells and Schwann cells. Satellite cells are like capsular cells which are also called as amphicytes. So, they cover the neurons like a capsule inside them. So, they are called as capsular cells or satellite cells, which are also called as amphicytes. And the next varieties are the Schwann cells, which are similar to oligodendrocytes, which form the myelin sheath around the axons of PNS, including cranial nerves from 3rd to 12th cranial nerves, and these cells develop from the neural crest cells. Peripheral nervous system is derived from the neural crest cells, so the Schwann cells are also derived from the neural crest cells. And each cell, we can see here, myelin forms a myelin sheath only of one axon. So, this is a Schwann cell. Structural division of nervous tissue. Can be divided as grey matter and white matter. Grey matter consists of nerve cell bodies, and white matter is mainly made up of axons. And apart from this grey matter and white matter, within the CNS, that is within the white matter, if there is a collection of nerve cell bodies, that is called as basal nuclei. And whereas in the peripheral nervous system, they are termed as ganglia. And white matter in the CNS forms tracts, ascending and descending tracts, and which are myelinated by oligodendrocytes. And whereas in the peripheral nervous system, they are myelinated by Schwann cells. Optic nerve, though we say it is a cranial nerve and part of peripheral nervous system, optic nerve and retina are considered as the component of central nervous system because the optic nerve is myelinated by oligodendrocytes. And if optic nerve is injured, it never regenerates, and because there are no Schwann cells. Apart from neuroglial cells and neurons, the peripheral nerve also consists of connective tissue. So the connective tissue which is covering the complete nerve bundles. So the outermost layer is the dense connective tissue which is called as epineurium. So this is the epineurium, and we can see the blood vessels and nerves traversing the epineurium, which supply the complete peripheral nerve. And apart from the epineurium, the layer of connective tissue which is surrounding each bundle of nerve fibers, which is called as nerve fascicle. So around each nerve fasciculus, there is a layer of connective tissue which is called as perineurium. Perineurium is clinically quite significant during microsurgery of limb salvage, means reconstructive surgeries of the limb. So during this procedure, this layer need to be reconnected for regrowth of nerve. The innermost layer which is surrounding each single nerve fiber is called as endoneurium. So the endoneurium is a thin layer of connective tissue which is wrapping around a single nerve fiber. Clinical significance of this endoneurium, it contains inflammatory infiltrate in Guillain-Barré syndrome. So this completes a basic introduction about organization of nervous system. Thank you.